This experiment uses biophysical techniques to assess the cross-linking capacity of the tomato cutin protective polymer and to monitor selective removal of epi and intracuticular waxes from fruit cuticles. The isolation of the cuticle from the whole fruit is achieved by enzymatic treatment of the tomato skins and adhering cell walls. In a second step, soxhlet extraction is used to de-wax the fruit cuticle exhaustively so as to obtain the cutin. Molecular characterization of the isolated cutin biopolyester is conducted by solid-state nuclear magnetic resonance. Solid-state NMR spectral analysis reveals that in cutins from wild-type and engineered tomato fruit cuticles, there are differences in the relative proportions of oxygenated aliphatic moieties that are capable of cross-linking. Alternatively, the whole fruit is painted and peeled with gum arabic to remove epicuticular waxes, and then dipped in chloroform to remove intracuticular waxes from the fruit cuticle. Then, the partially de-waxed cuticles, isolated by the same enzymatic treatment as the tomato skins, are examined using solid-state NMR and atomic force microscopy. Ultimately, the sequential removal of epi and intracuticular waxes is apparent through observation of a progressively diminishing wax peak in the NMR spectra and alterations in the surface profile of the fruit cuticle observed by concurrent AFM imaging.
Above-ground organs of plants are protected by a biopolyester-wax assembly called the cuticle. We will present protocols to evaluate the selective removal of internal and external waxes from tomatoes.